All right, why don't we go ahead and get started? Good morning. Thank you everyone for joining this lecture today. I'm Lawrence Lin. I'm the Director of External Relations and Outreach for the UCSF-Stanford Center of Excellence in Regulatory Science and Innovation, or CIRSE. Our CIRSE is a regulatory science center funded by the FDA with the goal of advancing regulatory science through collaborative research, education, and outreach activities, such as this lecture that you're listening to today. I encourage you to learn more about our center, the UCSF-Stanford CIRSE, on our website, which is ucsfstanfordcirse.org, shown in the bottom right of your screen. Before we start with today's presentation, I just wanted to give a plug for our upcoming Innovations in Regulatory Science Summit, which is scheduled for Sunday, January 10th, 2021. This summer, we'll focus on oncology, infectious disease, and the pandemic, and feature leaders from academia, industry, and FDA. Of particular note, we'll have keynote addresses from the directors of all three medical product review centers at the FDA. So that's Peter Marks in CBER, Jeff Shuren with CDRH, and Janet Woodcock at CDER. You can find the full agenda and speaker listing at the website shown on the screen, cirsesummit.com. And with that, I'd like to introduce today's speakers. So today we're actually going to have a number of people from the FDA give this lecture, so it won't be just one speaker. So I'll just briefly introduce all of them right now. Martin Ho is going to be our first speaker, and he's the Associate Director of Science for Patient Input and Real World Evidence in the Center of Biostatistics and Epidemiology at CBER, where he develops new tools to inform regulatory decisions, applies these tools to submissions, and builds review capacity at the FDA. Prior to moving to CBER, Martin was at the FDA's Medical Devices Center for 10 years and served as the founding director of the Quantitative Innovation Program, where he was responsible for evaluating novel sources of data to inform regulatory decisions. After Martin, Sarah Stothers will speak, and Sarah is a research fellow working on identifying innovative strategies for the inclusion of patient preference and experiences into the regulatory decision-making framework. Prior to joining CBER, she worked on patient and patient engagement initiatives with the Patient-Centered Outcomes Research Institute to advance patient-centered research. Sarah received her MPH and MSN degrees from Johns Hopkins. Joyce Lee is a research fellow working on patient engagement projects and a pilot natural history study for a rare disease. Prior to joining CBER, she worked in CDRH for 3 years on patient preferences, patient-reported outcomes, and real-world evidence. She has a master's degree in health economics and international health from Johns Hopkins. Hussein Ezzeldin currently co-leads the Biologics Effectiveness and Safety System C2, which focuses on development of new and innovative methods for automated reporting for CBER-regulated biological products. Hussein has worked on multiple risk assessment policy and research projects at FDA. He received his PhD in mechanical engineering from the University of Maryland and completed a postdoctoral fellowship at George Washington University. Finally, Xinyi Eng is a health economist and outcomes research scientist at CBER at the FDA. She has expertise in the elicitation of patient and physician preferences for medical products and using stated preference data to inform benefit risk assessments of health interventions. She received a PhD in pharmaceutical health science services research from the University of Maryland. And before I turn it over to Martin, who's going to start the lecture, I just wanted to give a little bit of logistics. We're currently using Zoom webinar, which is a little bit different than maybe some of the Zoom meetings that you're all familiar with. And so we'll be taking questions throughout the lecture, and we'll just kind of save them all for the very end when we have a Q&A session. In order to send a question, click on the Q&A button that you'll see in the middle of the bottom of the Zoom window. So you click on that Q&A button, and then that's how you can submit a question. With that, I'll turn it over to Martin to start the presentation.
Thank you, Lawrence. Joyce, can you Yes, thank you, Joyce. So thank you very much for having us this morning. Excuse me. Um, Today we are going to want to talk about a vision that we have in terms of patients being in the center of driving multiple fronts of innovations in the clinical trial enterprise, and especially in considering the benefit risk assessment of products. Next slide, please. So I'm bringing in a multiple of my colleagues in order to basically convey a very consistent message that by putting different pieces together, we would be able to get a much better, picture, not only larger but more complete picture of how we can make use of these innovations at multiple fronts to help us to make better decisions. First, I will talk about how these different areas intersect, and they all overlap on one thing, which is the patient as a role, as a pivotal role in driving all of this. Then next, Sarah would introduce a three patient preferences study that is being sponsored by CBER, specifically designed to address some of the benefit risk problems that we are facing when we are evaluating innovative um, medical products that have a categorically different benefit risk profile than the existing treatment. Then Joyce will talk another perspective of how to approach this issue by um, talking about our pilot study to leverage natural history study to tackle to tackle one of the problems of rare disease, randomized clinical trials. It's very important because it's not only the disease that the natural study is targeting, but I think it's quite common that they are facing a recruitment barriers. And this pilot study will be an important first step for us to explore possible solutions. And to supplement all of the areas that we mentioned by Sarah and Joyce is an app that CBER developed from scratch to support patient-centric data collection. And hopefully it will shed some light on how we can conduct siteless clinical studies. So let's start on the first part of the story, which is um, we believe that as patients gaining more, feeling more empowered, they are very eager to participate in more of the healthcare that they are receiving, and the regulatory portion of the healthcare is of course part of that. And FDA has been very driven to play a proactive role in listening and in measuring even the patient's input, and that will in and that's how we are adapting their input in several different areas. Um, you may have heard that recently the FDA has established a digital health center of excellence, which is a cross-cutting body across all the medical product offices. And I will also talk a little bit about a real-world data program that we have recently established. And of course, Silas, RCT is in everybody's mind, especially in this COVID-19 situation. And we definitely have learned quite a bit of lessons from that. However, last but not least, I mean, the rare diseases has always been on our staff at Center for Biologics, mostly because I think within the three medical product centers, CBER has more rare diseases in hand, and I think we also believe that some innovative and treatments such as gene therapy would hold the key to address a few of those that had been regarded as incurable. But I would say that underlying all this effort is helping us to collect data and use the data to generate evidence for us to, as a regulatory body, to assess the benefit risk ratio of individual products. Next slide, please. So why do we want to collect patient input? In addition to the fact that it's the right thing to do, we are also have a mission for the FDA to not only protect, but also promote public health. And by collecting their input, we believe that it's crucial for us to increase the therapy access to a few populations, such as a vulnerable population, where diseased patients, pregnant women, immunocompromised persons, and perhaps we can even identify appropriate cohorts by through clinical diagnosis. And Center for Biologics is a great center for us to consider that, mostly because we have seen increasing number of gene therapies coming in our center, and they all share a common feature, which is they carry high rewards, such as potential cure, but categorically different types of risk, for a number of disease, such as cytokine release syndrome, that's inevitably associated with gene therapy for some. And last but not least, because of the innovative nature of these technologies, the length of the history and also the information, comparatively speaking, have a larger uncertainty. Not only that, not only we don't know some of the parameters, but there may be some unknown unknowns, to quote Don Rumsfeld on that. So we have to make sensible and reasonable decision in order to be a good and smart regulator. And we are talking about that in the context of facing unmet medical needs and especially dire conditions. So in those situations, there's a situation what we call a preference-sensitive decision making, which basically, simply put, is that in order to make improvement in benefit, someone has to give up to tolerate an increasing number of increasing amount of risk or different type of risk. So in other words, there is no free lunch. If we want to have a better outcome, we may need to tolerate a new type of risk or more risk than the existing treatment. So I think that is a quantitative assessment or characterization of of the situation that we just mentioned about a high risk and high return situation. Next slide, please. And to highlight the role of patients' input in regulations, there's a series of legislation that confirm that. And in fact, I think specifically in the Precision Medicine Initiative is specifically stated that the ideal treatment is personalized to both ourselves and our cells. So in other words, they're not only working at a biological level, but it should also provide benefit at our daily life level. Next slide, please. And then next slide. So here, during the 21st Century Cures Act, here I want to highlight a section which specifically asks the FDA to make our consideration of patient experience data submitted in our approval decision of the new drug application, which is NDA or BLA, biologics licensing application and or approval. And so I think this is a very big breakthrough from my perspective, because for the first time, the patient's effort and patient's experience has been formally mentioned and stated in a, in a government documentary documents about the patient's own perspective on getting receiving the medical products. Next slide, please. So in response to that, our center has established a science of patient input program. And this program, our mission is to advance the science of patient input to help inform our regulatory decision making and policy development, to help us make better decisions and more patient-centric policy development. For example, for clinical trial design, we can definitely use patients' inputs in recognizing the relative importance of the endpoint and how it's being developed, and trying to size the probably power the study by looking at what is what we call clinical meaningful difference between treatment and control. And more importantly, there would give us more context about the relative importance of type 1 versus type 2 error. In other words, the relative cost of committing false positive versus false negatives really depends on the context. For situation where there's no existing treatment for a dire condition, the price of false negative, the price of false negative is much higher than a false positive. But in a situation where we have many Me Too drugs or Me Too, medication, in that case, I think the relative cost of the false negative is much less or much less costly than the false positive, because we already have some medications with a similar efficacy. And again, these are in the relative terms. And similarly, in benefit risk assessment, we have pre-market licensing and post-market surveillance. Both can definitely can leverage the patient's input. Next slide, please. So here is our team at Center of Biologics under the Office of Biostatistics and Epidemiology, and you will hear most of them speak after this, after me. So next slide, please. So now let's talk a little bit about real-world data and real-world evidence. For me, I think it's really amazing to see how the real-world data definition evolved for the last 3 to 4 years. And this is a definition that is captured by, from the latest documents that we have on the topic. And basically, what I wanted to highlight here is in terms of real-world data, in addition to the usual suspects of electronic health records and claims and bills data, we also include the patient generated for the first time in home use settings, and the data generated from other sources that can inform health status, such as mobile devices. And real-world evidence is basically a purposeful, in a designed manner to derive the evidence based on the real-world data. So as we are expanding the scope of real-world data, I'm very happy to see that more and more new sources of data has become available. So I think this is definitely a very promising area. Next slide, please. In 2019, December, our agency has released a real-world evidence program framework, and the framework lays out 3 different criteria. The first one is the characterization or evaluation of real-world data, whether these data are fit for use in for a specific regulatory decision. And then next is the study design, whether the way that we collect this data and analyze them would generate adequate scientific evidence to address the regulatory decision in hand. Last but not least, does the study being conducted, in a way that meeting with the FDA's regulatory requirements. Next slide, please. So in order to bring this back to the benefit risk assessment context, perhaps it was helpful for us to think about, to be more specific about what we mean by benefit risk assessment. And the best document for me to use to introduce the topic is the guidance that is being released, which is a final guidance released on 2016, is a guidance on benefit on factors to consider for benefit risk assessment on medical devices. Is being jointly released by Center for Devices and Center for Biologics. Next slide, please. So first of all, the factors that we will be considering often are the two usual suspects, benefits and risk. But what we need to be a bit more, careful is first, benefits and risk have multiple dimensions, such as the type and the magnitude, the probabilities, and the duration. But moreover, for risk, because it's medical devices, it includes also the diagnostic or screening tests. So the false positive and the false negative rates should also be considered. But these benefits and risk do not exist independent of its context. So where do we define the context? Next, please. The context is actually defined by additional factors as follows. Here is a list of 7 of them. I wanted to highlight perhaps 2 of them, which are likely to be able for us to quantify them. The first is the uncertainty of the evidence that we have, and the second one is the patient's tolerance for risk and perspective on benefit. Next slide, please. So here, the guidance says that we should treat the patient's tolerance for risk and positive on benefit as evidence. And here in the FDA, evidence has a very specific legislative definition. And so therefore, in order to elevate the measurement that we gathered to measure the patient perspective, we have to meet certain standards. And even though we know that such a requirement, but we don't actually know how to, gather those information, what we call patient preference information, how to design such a study, how to evaluate them. And needless to say, how to submit them to the agency for consideration in support of their medical product submissions. So next slide, please. So next, I'm going to turn it over to Sarah, who is going to tell you about how we can measure this, these patient preference information in a quantitative manner. So thank you, Sarah.
Thank you, Martin. I just want to make sure everyone can hear me.
Yes.
Thank you. Good afternoon, everyone, or I guess it's good morning on the West Coast. My name is Sarah. As Martin mentioned, I'll be talking about some of the patient preference studies that CBER has sponsored. As Martin had mentioned, patients may be willing to tolerate higher risks in exchange for better efficacy. For the purposes of studying this, we decided to choose 3 areas or conditions to understand how patients weigh the benefits and risks. So we chose hard to control type 1 diabetes, which we're collaborating with UCSF and The second project is sickle cell disease, which we're doing in collaboration with Duke. And the third patient preference study is osteoarthritis of the knee that we are collaborating with RTI on. Next slide, please. So the first PPI study I want to talk about is the hard to control type 1 diabetes. A lot of patients who have type 1 diabetes experience difficulty managing their blood glucose despite optimal insulin therapy and can experience something called hypoglycemic unawareness. Usually when patients experience hypoglycemia, it may come with symptoms such as nausea, vomiting, and loss of consciousness. Sometimes hypoglycemia can happen without these symptoms. And research has shown that volatile fluctuations in blood sugar control can contribute to poor longer-term outcomes. Islet cell transplantation is a treatment strategy for patients with hard to control type 1 diabetes that may help control this and not cause such severe volatility in blood sugar control. You'll hear this more commonly referred to as brittle, which is a lay term that is used for type 1 diabetes. Next slide, please. Thank you. So a couple questions we wanted to ask about islet cell therapy, especially when it comes to patient preferences, is how do patients weigh the benefit-risk trade-off for islet cell therapy? The second question we wanted to answer was, what attributes have the greatest relative importance on influencing patients' treatment preferences? And finally, how do these findings inform regulatory decision-making? Next slide, please. So just to orient a few, some people may not be familiar with patient preference studies, but we use something called discrete choice experiments. This is a well-established methodology that's actually borrowed from economics, and it's used to elicit and quantify preferences on healthcare products and interventions. So respondents choose between hypothetical treatment profiles. Each profile is described in terms of a number of characteristics, or what we more commonly refer to as attributes that can be related to efficacy, safety, route of administration, and benefits and risks. So you can see an example in the diagram in the right. You can see at the left-hand side of the diagram, you'll see attributes listed out, which can be, again, the route of administration, benefits and risks of the treatments. And then you'll see on the right-hand side of the diagram, treatment A and treatment B, which are the two different treatment profiles. And under those, you'll see the levels, which are variations of the attributes or different amounts of benefits and risks related to those attributes. This is a sample choice task that a participant will be presented with, and they'll be able to select their preferred treatment based on this profile. Now, this is one example. Most participants will receive 12 to 18 questions that look like this, or we call them pairwise profiles. Next slide, please. So for the purpose of the type 1 diabetes islet cell therapy study, we selected 8 attributes, which you'll see listed on the left side of the table. For example, chance of achieving clinical treatment success. We also have defined them on the right side of the table for purposes that respondents can understand. And I'll just pause here for a few seconds just to give people an opportunity to look through the attributes. Next slide, please. So based on these attributes, and then we select levels, corresponding levels, which you can see in the table to the left. I've only highlighted 4 attributes, not all 8, just for the sake of space on the slide, so you could properly read them. But you can see that we've translated the attribute and the corresponding levels and varied them to create a question or choice task. Now you can see the attributes listed on the left side, and then option 1 is treatment profile 1, essentially, and option 2 is treatment profile 2. And then the levels are randomized and inputted into the treatment profile. And participants are faced with this choice task and asked to select which one they would most prefer. Now, this is just one example. Again, the participant would probably receive 12 to 15 of these kinds of choice tasks in a survey format. Next slide, please. So we did this on about 92 participants were included in this survey. We did a convenience sampling of UCSF diabetes clinics and national diabetes research centers. The participants that were included were adults 18 years and older who were English speaking because the survey instrument was in English, and we wanted to make sure that people could comprehend the questions being asked. These were physician-referred type 1 diabetics who previously experienced a hypoglycemic episode. And the nice thing was, because we recruited from clinics, we knew that they were absolutely physician-referred and not self-reported. And you can see the patient characteristics listed in the right-hand table. Our sample is predominantly white. You can see their education level and the type of health insurance. Next slide, please. So we received results based off of the sample of 92. Just to orient you to what you're seeing here on the slide, this is a representation of the preference weights for all attributes. You can see the attributes listed, all 8 of them listed at the top of the figure. And then you can see the corresponding levels on the bottom hand, or the x-axis of the figure, and then the preference weights on the y-axis. Next slide, please. You can see better outcomes have significantly larger scores, which is indicated by the green box with the green arrows. And then worser outcomes has significantly lower scores, which you can see exemplified by the red box with the red arrows. The nice thing about this is we can compare attributes against each other to see where patients have hold more weight. And you can compare levels within attributes or across attributes. Next slide, please. So an example of this is we can look at the most important attributes, which are listed by the yellow boxes and those weights, but we can also compare those weights. So for instance, we can take serious complications and the height of 0 to 15% and compare it to treatment success of 40 to 90%. Next slide, please. And compare the height and see that the serious complications of 0 to 15% is most important when compared to treatment success of 40 to 90% for participants. Next slide, please. Thank you. We can see this in a different depiction by using a bar graph, looking at the relative importance of attributes. So again, you can see serious complications towards the bottom of this figure of 0 to 15% and see that it holds greater importance than chance of treatment success of 40 to 90%. And this is just a different way to kind of look at the relative importance of the individual attributes against one another. Next slide, please. So some of the key findings from this study that we noted were the 3 most influential attributes to patients' choices for islet cell treatments are what is their risk of experiencing serious complications? And we noticed that from 0 to 15% held the greatest importance. Secondly, we found that independence from insulin and improvement from never to 5 years held the second greatest importance. And how long treatment success lasts, improvements from 0 to 0.5 to 5 years. And this study also helped to demonstrate that patients are willing to make benefit-risk trade-offs when choosing islet cell treatments. And this can help inform regulatory considerations of islet cell treatments by attaching patients' preference weights to the outcomes observed in the actual clinical studies, which can help reviewers when making clinical decisions. Now I'll move to the second patient preference study that we did, which was osteoarthritis of the knee. Osteoarthritis is characterized by degradation of knee cartilage and bone. Patients who have osteoarthritis of the knee can experience increasing pain and functional impairments. And while there are available treatments, most of these offer symptomatic relief and don't slow the progression or affect the pathophysiology of the actual condition. Some of the newer therapies, such as cell therapies or gene therapies, actually may affect the pathophysiology and actually help to slow the progression of OA. So one of the questions we had, next slide please, is what is the relative value of improvements in pain versus function to patients? Next slide, please. So for this study, we selected 4 attributes that represent benefits and risks. You can see the attributes listed on the left side of the screen. Improvement in pain, improvement in function, how long improvement lasts, risk of developing too much tissue inside the knee, and then attached corresponding levels to each of these attributes. Next slide, please. Then we took these attributes and their levels and made treatment profiles. You can see that the treatment feature corresponds with the selected attributes, and you can see this on the left hand of this diagram. And then the varying levels can create different treatment profiles. So you'll see treatment A, treatment B, and then no new treatment or the standard of care. And participants are faced with this choice task and asked to select which profile they would select. Now, this is just one example of many questions that a participant would receive in the survey instrument. Next slide, please. One of the key things that I want to point out about this patient preference study is that one of the more innovative ways that we did this was we took a patient-reported outcome measure tool and adapted this to make our PPI attributes. One of the more common clinical outcome assessments used in clinical trials for knee osteoarthritis is the Western Ontario and McMaster University's Osteoarthritis Index, which is more commonly referred to as the WOMAC. And the WOMAC is made up of 3 domains. Oh. Give us 1 second. Thank you. You can see that on the right-hand side of the screen is an example of the WOMAC, and it has 3 domains: pain, stiffness, and function. We selected pain and function for the purposes of this study because they were found to be correlated to the treatment benefit. Stiffness was not. And you can see, if you click again on the slide, thank you, you can see that we selected the pain domain and the function domain. Within each of these domains, there's 5 items that the respondent is asked to answer. And in the function domain, there's 17 items. What we were interested in doing was creating 2 choice experiments to facilitate the comparison between the total domain score of adding up all those items in each domain, and then comparing that to a single item score version, so selecting 1 item from each of those domains. The single item that we selected was walking on a flat surface because it's in both the pain and function domain. Next slide, please. The final study I'd like to present today is the sickle cell disease patient preference study. There are a lot of gene therapies coming out that offer a potential cure for sickle cell disease, but they pose a lot of high risks for patients. So what we were interested in learning was how patients make the trade-offs between these benefits and risks, and do patients with more severe symptoms view the benefit-risk trade-offs differently than those who may have less severe symptoms. Next slide, please. So with this PPI study, we selected several attributes, 4, which are listed on the left side of this table. These include chance of no symptoms of sickle cell disease, increase in life years, the chance of dying within the first year after treatment, and increase in lifetime risk of cancer, and selected attribute levels for each of these attributes. Next slide, please. So here's a sickle disease study question from the actual survey. So you can see the attributes that we selected, the 4, on the left side of the screen. And then the varying levels are randomized and create these treatment profiles. So you can see treatment profile for gene therapy A, and treatment profile for gene therapy B, and then standard of care, no gene therapy. And participants will select their preference based on these differing profiles. Next slide, please. So what are some of the PPI contributions that we've learned? Well, we acknowledge that patient preference information is important, and it's important to complement clinical and statistical evidence to make these benefit risk assessments. These 3 studies provide additional evidence on patient preferences and that they can be scientifically obtained and quantitatively measured. We can also, patient preference information also provides insights to reviewers, clinical reviewers at the FDA, who may have limited experience with patients of some rare diseases, and understanding that patients may accept more risk for a particular benefit. And I do want to note that the science of patient input is constantly evolving. A good example of this is when we used a PROM and adapted this to create PPI attributes, and finding ways to innovate this kind of work. Next, I will turn it over to my colleague, Joyce, who's going to talk about a natural history study for a particular rare condition.
Thanks, Sarah. So the next project we want to share about is the natural history of the metachromatic leukodystrophy, or MLD, the home study. And to begin, we know that randomized controlled trials, RCTs, are often regarded as the golden standard for drug development. However, RCTs may be more difficult for the rare disease community and population. And that is because what if for a rare disease participant in a randomized control trial, what if they were randomized to a control trial? Or what if they become ineligible for another clinical trial because they're participating in this trial? And especially with rare, fast progressing diseases, how is that going to work out if they are in a randomized control trial that would take many years to participate in? So to answer these questions and to explore potential solutions, we are trying to use the natural history study to explore how we can address these issues for the rare disease population. So what is a natural history study? A natural history study follows a group of people over time, and it collects health information to help us understand how the disease or the condition develops over time and how we can potentially treat it. So in 2019, the FDA published a draft guidance on the natural history study for drug development for rare diseases. And that is exactly what we're trying to do with our study that I want to share with you today. We're trying to develop a pilot to learn how to use an NHS to be used as an external control for future RCTs. To do that, we collaborated with the National Organization for Rare Disorders, NORD, and IBM on this project. The disease we selected, MLD, is a rare disease that does need an external control for single-arm trials, and there is potentially products that may be coming for submission in the next few years. MLD is a rare progressive disease. The main symptoms include difficulty walking and talking. There may be seizures and also personality and behavioral changes. The cause is usually seems like a genetic cause, and the main population or the majority of the patient population lies in the late infantile stage. So children or toddlers under 3 years old, that's where the majority of the population is. And there are less cases of juvenile and adult MLD cases as well. So I want to highlight three significances of the project and three innovations that we really are tackling with this pilot. The first is the use of a multi-stakeholder approach. The second is learning about the best practices for designing a natural history study. And the third is using innovative data collection methods to mitigate drawbacks of traditional natural history studies. And on this point, Hussein will also talk more about this once we get to that point. So the first one is our collaboration with multi-stakeholders. We have worked closely with the product review office and the FDA, the reviewers, and also NORD and patient groups. We participated in monthly advisory group meetings that is facilitated by NORD. We have input from patients and caregivers and clinical experts to design the study for regulatory decision-making. Also really trying to be patient-centric and to promote patient-focused research and product development. And as part of that, one of the ways we are collecting data in the natural history study is through a sightless video assessment with the patients and caregivers. And in this process, the study coordinator, who has a clinical background, can answer any questions the families may have and support them along the way. We're hoping that this would have less patient burden on the families, and also there may be more accuracy in data collection and would reduce missing data. And we're also looking for timely feedback, and we can just check in with the families in each of these video assessments we conduct. The second significance of this project is understanding how to design and conduct a natural history study to serve as an external control. because currently there is not much information at all about how we would do this. We also want to learn how to combine and analyze data from this study and how to control for confounding and biases along the study. This is also an opportunity for us to incorporate CBER's clinical data requirements in a longitudinal study. This is what the NORD's pilot homepage looks like. It is already launched, and people who are interested can join the study via this page. The third key significance of this project is the innovative data collection methods. We will use a mobile and web-based app to collect longitudinal data and patient perspectives on their disease status and potential treatments. And here I will pass it on to my colleague Hussein, who will share more about what this looks like and what this app is all about. Hussein?
Thank you so much, Joyce. Good morning, everyone. Next slide, please. Good morning, everyone. I'll be speaking today about the Biologics Effectiveness and Safety Initiative, which we call BEST for short, a SHAPE app. As you can see here on the slide, this is the landing page for our mobile and web-based app called SHAPE, short for Study of Health and Patient Experience app. We decided to develop this app because we recognize the need that being able to incorporate patients' input, we needed a tool, a flexible tool, a versatile tool that would allow us to collect their input in many different ways. So as you can see here, we wanted to collect various endpoints. Some of them would be scheduled endpoints, for example, the gross motor function classification, which is a primary endpoint in most of the MLD clinical studies. We also wanted to have the flexibility to collect unscheduled endpoints, mainly adverse events and relevant health events. We also wanted to achieve with this app, increasing the compliance of the data, reducing missing data. This is one of the big challenges that most of the natural history studies or registries face when you are collecting longitudinal data for over a period of time. And we hoped bringing this platform to people's mobile phones will reduce missing data. Another goal of developing this app and using it is to be able to compare the clinical or the caregiver reported outcome together. So, as Joyce alluded, in our study, the clinical coordinator or the study coordinator is going to conduct video assessments at several points during the study. We wanted to compare this assessment from the clinical coordinator to the caregiver reported assessment using the mobile app. The other goal of this app is exploring linking EHR for our study patients or our study participants. The main goal of doing something like this is leveraging the advancement in EHRs, advancement in artificial intelligence. We can lessen the burden on patients to report changes in their health conditions, report changes in the medications. Having this access, we can mine the EHRs, collect this data, and have more accurate information about disease progression and changes in their conditions. Next slide, please. What I'm going to try to do now is I'm going to try to give you a very brief demo of how this app is structured, what it offers, and what are the capabilities and strength of this app. You see here on the left, this is the landing page of the app. It has at the bottom 4 tabs. One of them is for the survey, which is the far left. The one on the far right is for the diary entries, which is allows patient to or participants to report any health event or withdraw from the app, or even report a clinical visit to their doctors. In the middle, there are 2 apps to allow them to create their profile, add participants or add patients to their profile, and also receive messages from the study coordinator. You can see here in this demo, this particular participant in the study had 2 surveys deployed to them. The one at the top is the MLD, gross motor skill at 90 days. And the one at the bottom is the one for baseline. Next slide, please. The second tab, which is the profile, allows the person using the app, which we call respondent. This person can add participants. So usually the respondent would be the caregiver, a parent or someone else. And then they can add multiple participants, which are patients, to the study, to whom they can provide either response for surveys, or they can report also health events. Next slide, please. And on this slide, I'm showing you the diary page. And as I mentioned, we had the survey capability or functionality to allow one way of communication from the study coordinator to the patients. The diary tab or the diary option allows the second way of communication, which is if the patient or the participants wants to report any health event or doctor visit, or even withdraw from the study, they can use this tab to accomplish this. On the left here, you can see that if they go to this tab, they can select the survey. And I say here survey/study because our app or our platform allows for multiple studies. And by this, I mean you can start from a specific cohort of patients that join your study. And then if you're interested, you're able to create other studies which might be trying to explore specific parts of the disease or specific treatments or specific cohort or subcohort that is enrolled in the bigger study. So if someone is using the diary tab, they would select which study they are reporting this for. They would select the participant, and then they would select what kind of event they would like to report. Next slide, please. And when they select the type of study, or they select the study, select the participant, and what kind of health event or event type, they say that they can go now into providing more details. And in this example, you can see here the page for reporting a health event. You can provide, you can pick from other things. And in this case, I believe I chose an ER visit. So you can provide more information about this event. You can report the type of outcome that happened, whether you recovered or you are still recovering, or this event was fatal. You can also provide information about the treatment that went during this event or even after this event. You can provide all this information. And if I go to next slide, you can see here that this becomes an entry on your diary page or your diary tab. And if you click on this entry, you get all the information that you provided. So it's there for your record, and also the study coordinator can download this information. So by this, you allow for the study coordinator to collect data at specific time points, but also allow participants and respondents to provide information at any point during the study. And this makes this process very seamless and easy. And by this, I will hand it over to Martin to give us the recap and the takeaway. Oh, I have a couple more slides. So what I'm showing here is this is the general layout of how this app fits into the NORD home study, you have the patient is interacting with the Shape app through their mobile phone or a web browser. They provide information to the natural history study, and this information can go to a clinical trial to augment this clinical trial and provide information about external controls. And this will feed into regulatory decision-making. This is not the only way that the patient can interact with the study. As we mentioned at the beginning, we also have the capability of allowing the patient to provide access to their EHRs through their healthcare systems. And this information also will go to our clinical trial design and inform our regulatory decision-making. Next slide, please. And as I mentioned here, the study involves NORD or CBER side to create surveys and deploy them. And this gets notifies the patients or the participants of the study. They are able to access the app or through their mobile phones or a web browser, provide the answers to these surveys, or alternatively, they can also provide diary entries. This goes to NORD, which stores the data of the surveys and the diary entries in a secure database, and then conduct analysis on the responses that they were able to download, and then inform CBER with the results of this analysis. And I believe this is the correct point to hand it over to Martin. Thank you so much.
Yeah, thank you, Hussein. Yes, thank you, Hussein. I just want to say that thank you very much for listening to us. And I hope that during the last couple minutes, a few minutes that we would like to introduce to provide you with a glimpse of multifaceted research interest from Center for Biologics about patients, including quantitatively measure their preferences, using natural history study to augment a potential concurrent control for rare disease clinical trials, and needless to say, also the development of an app for us to explore different ways of conducting clinical trial enterprise. So here the takeaway messages is we believe that the patient's input playing an increasingly important role for our assessment of the benefit risk benefit risk ratio of the individual products, and also in enhancing the clinical trial enterprise. And the emerging breakthroughs of biological products provide us with very strong motivation to keep on developing and exploring these types of new tools for us to tackle the challenges of benefit risk assessment. And we believe that by measuring patients quantitatively, we can supplement the information that we already have on the clinical evidence to help us to tackle these problems. And last but not least, I think I just want to make a call of action and invite you to work with us to collaborate on bringing these novel innovations into the 21st century's clinical trials enterprise and benefit risk assessment. Thank you very much.
All right, thanks to the FDA team. We unfortunately don't have time for questions, but I saw that the FDA team has been answering the questions via chat. And so if you have any other questions, please get in touch with us and we can forward those questions to the FDA. This presentation also has been recorded and will be posted on the UCSF-Stanford CIRSE website soon. All right, thanks everyone for joining, and we'll see you next time. Thanks.